Welcome to the Bio 341 lecture on genetic drift, effective population size, and neutral theory. We've already covered two of the four forces of evolution, the first being mutation, the second being natural selection, and now we're covering genetic drift. Then later on we'll cover gene flow. So think about a system that's in Hardy-Weinberg equilibrium. We have a blue fish, it is a homozygote for the B allele, a yellow fish, homozygous for the Y allele, and a green fish, which is a heterozygote, a mixture of blue and yellow. The population meets all the assumptions of Hardy-Weinberg, and we have a population of 10 fish, and suddenly there's a mutation in one of them. And the mutation is from a B to a Y. And it's a single mutation, so it changes one fish from a homozygote to a heterozygote. What is the new frequency of the Y allele in this population? Stop and calculate this for a moment. You should have come up with, there are 20 alleles in the population because there are 10 fish. There's one Y allele. So one allele out of 20 is 0.05 frequency in the population. And any new allele that's introduced by mutation into a population will typically have an allele frequency of 1 over 2 times n, where n is the size of the population. So 1 over 2n for a nuclear gene. Remember, though, that alleles can be introduced into a population through immigration. So new alleles don't always have to arrive due to mutation. So, genetic drift. We can define genetic drift as the random stochastic changes in the frequencies of 2 or more alleles or genotypes in a population. And this is just caused by basically sampling error. The actual physical processes that cause this are all based on the fact that population sizes are finite. There's only so many individuals in a population, and different genes will have different reproductive success simply due to stochastic events. So the first category of things that contribute to genetic drift are Mendelian segregation and independent assortment. So each gamete will only contain one copy of the parental genes, and which copy of each gene is in that gamete is a pretty much random process due to recombination. And then there's random sampling of those gametes. So which male and female mate, that can sometimes be a selective process, but there's also randomness involved. Which sperm meets with which egg, that's a fairly random process as well. Finally, there can be large stochastic shifts in population sizes, where you have a huge population decrease, for example, but it's a large-scale change, but it is a random, it picks at random who is killed off. So this is essentially a random event. All of these things contribute to sampling error in the population, and therefore not all of a parent's genes will be passed along to their offspring. So if you think about sampling error in science, when you're doing an experiment, it's pretty intuitive that the more samples you have, the better you understand the underlying distribution, the underlying thing that you're trying to figure out. So if you're trying to figure out whether a coin is fair, and you flip it 3 times, you'll get a head to tails ratio of 2 to 1. Now, nobody would claim based on that data that the coin was unfair, that it flipped twice as many heads as tails. We would all acknowledge that that's just a matter of undersampling. We haven't sampled enough to get a good ratio. If we flip the coin 300,000 times, we're going to get a ratio that's very close to 1 to 1. This is not because the probability of head or tails is different for this coin. It's a 50-50 coin, but it's because of sample size, and that's the same concept that underlies genetic drift. So, based on this concept, it's fairly intuitive then that small populations are basically small sample sizes of the alleles in that population. And since they are small sample sizes, they are more prone to large shifts in gene frequencies due to this random sampling error. So here's one way to think about it. Think about a large jar full of alleles, and it's just a 2-allele system. It's either red or black. And I go in and I pick out these individuals to reproduce. Well, in doing so, I've picked out 1, 2, 3, 4, 5, 6, 7, 8 individuals with black alleles, and 1, 2, 3, 4 alleles with red alleles. So the frequency of the red allele in this population is 0.33, which is 4 out of 12. Now, the true allele frequency in the entire population is 0.5 and 0.5, but just based on my random sample, I've completely changed the allele frequencies that I sampled. And so if we do this multiple times, if I reach into this jar and accidentally pick more blue alleles to reproduce than red alleles, then I'll have more blue alleles in the next population. And if I reach into the jar again and pick, I'll tend to pick more blue alleles yet again, and end up with even more blue alleles in the next generation. If I reach into this jar, okay, now I stochastically get a few more red alleles in the next generation, but finally I end up removing all the red alleles from the population simply by randomly reaching into the jar. So, some terminology. Polymorphism. A locus with more than one allele is polymorphic. And heterozygosity. Heterozygosity is the frequency of heterozygotes in a population. And this is the way that we estimate genetic diversity in a population. So we can divide this into two different concepts. The first is expected heterozygosity. Expected heterozygosity is the heterozygosity that we expect given Hardy-Weinberg equilibrium. And it's simply based on 2pq. Then the observed heterozygosity is the actual frequency of heterozygotes in the population. So we count up the number of heterozygotes and divide by the total population size. That is our observed heterozygosity. So for a population with 50 big A, 10 big A little a, and 40 big A little a, what is the expected heterozygosity and what is the actual heterozygosity? Okay, so the first thing we do as always is figure out how many individuals we have in the population. In this case, that's easy. It's 100 individuals, and they each contribute 2 alleles to the population, so we have 200 alleles in the population. Okay, now we want to figure out P is the frequency of big A is equal to 2 times each of the homozygotes plus 10 over 200 is equal to 0.55. Q is equal to 1 minus 0.55 to 0.45, and 2pq is equal to 0.55 times 0.45 times 2 equal to 0.495. And this is the expected heterozygosity. Then the number of heterozygous individuals in the population, there's 10 of them out of 100, so 0.1. This is the observed heterozygosity. So if we go back to our little population of fish, and we have our one green mutant, and we want to calculate the genotypic frequencies, we know that the frequency of BB is 9 out of 10, 0.9. Frequency of BY is 1 out of 10, 0.1. Frequency of YY is 0. There's no yellowfish in the population. From this, you should be able to calculate the allele frequencies as well as the expected and observed heterozygosity. Give it a shot now. Okay, the allele frequencies out of this, 19 out of 20, for B, 1 out of 20 for Y, and the expected heterozygosity 2pq is 0.095. The observed heterozygosity is right here at 0.1. So, under the assumptions of the Hardy-Weinberg equilibrium, a new allele will stay at its initial frequency, because the assumptions are that we have infinite population size, so all alleles are represented in the next population. In the real world, the frequency of this will change stochastically due to genetic drift. And an allele will either go to fixation, meaning that p will equal 100%, or it'll go to loss, or p equals 0. So let's take a look at this in allele A1. If we start off with an allele frequency of 0.01, so this is a new allele, and we take it, we let that run, we see the allele frequency doesn't change. If we change the population size down to something like 10 and decrease our x-axis, now what is the probability that this will go to fixation? It's actually the same as its initial frequency, because only one allele can go to fixation, and the one that starts at 0.01 has a 1 in 100 chance of going to fixation. So we'll see whether we can get any of these alleles to go to fixation. Oh, there, one went. But most of them are going to go extinct. Now, if we change the effective population size to 100, still most of them are going to go extinct. What if we change the initial frequency to 0.5? Now this is simulating an allele that's already present and very common in the population. So let's run that through genetic drift. And you can see that at effective population size of 100, most of the variation stays inside the population. If we expand our y-axis to 1,000 generations, though, we'll find that most of the alleles either go to fixation or extinction. Within about 200 generations. Oh, that one's stuck around for a while. Okay, what if we change the population size to 1,000? Do that, we're going to see that the alleles have a much greater chance of staying in the population. So they're staying a lot closer to their initial frequency. Okay, that one went to fixation. But they're staying closer to 0.5. And if we turn it up to 10,000, then they're going to stay really close to their initial frequency. As we get closer and closer to infinity, we'll get closer and closer to a straight line. So genetic drift is very strong when population size is very small. It allows the allele frequencies to vary a whole lot. But when population size gets large and sample size basically gets large, we reduce that noise in the population, and we end up with lines that are fairly straight and don't vary all that often. So, in small populations, starting at allele frequency of 0.5, small populations will lose a lot of their genetic variation, whereas large populations will keep their genetic variation. All the alleles will stay at roughly at similar frequencies. And so small populations have high genetic drift, large populations have low genetic drift. So we return to our original question. We have this one individual that is mutated from a B allele to a Y allele, such that it is a heterozygote that is green. And we can think about this in terms of DNA, where we have a mutation, a non-synonymous mutation at this base pair that changes from an A to a G, and so this would be our Y allele, and this would be our B allele. And we ask this question, what is the fate of a new allele? Well, there's really only two fates. One would be extinction, and that's the most frequent fate, especially for new alleles. Their probability of going to fixation is very low, and so they'll most likely go extinct and be removed from the population. However, the other possibility is that it could go to fixation, in which case this Y allele would completely take over the population. In terms of DNA, when we find that an allele has gone to fixation, we call this a substitution. So this A has completely been substituted for a G and is now completely fixed in the population, this G. However, on the way to fixation, in between extinction and fixation, there exists polymorphism, where you have both alleles segregating in the population. Although one of them is definitely on its way to extinction, we don't know which one is on the way to extinction, especially under a genetic drift scenario. And so we end up with this measurable polymorphism in the population. So mutation is a change in the DNA of a single individual. A substitution only occurs once that mutation becomes fixed within a population or species. And a polymorphism is a locus with more than one allele that is segregating in the population. Now, this concept or conceived of by both Sewell Wright and R.A. Fisher in what's called a Wright-Fisher population model. In a Wright-Fisher population model, we have a population of individuals that are hermaphroditic. They have a constant population size. They're semiparous, which means that they die as soon as they reproduce. And they produce lots and lots of gametes, of which only a few are randomly successful. And we can take these individuals, and only some of the individuals are going to be able to successfully reproduce. This individual produced 2 offspring, this individual produced 1, this individual produced 0 offspring, and so on. And we can simulate that into the future, and this is one of the most classic population genetic models that exists. So we will do this in the classroom. We will simulate this to give you an appreciation for how genetic drift works in a population. So in a small population, genetic drift is very strong, and it removes genetic variation. It causes genes to either go to fixation or extinction. And therefore, at a population size of 4, heterozygosity is lost very, very quickly. In a population size of 40, heterozygosity is lost a little bit less quickly, and a population size of 400, heterozygosity is lost, is just barely lost from the population. And there's an equation that allows us to predict this at h at g plus 1, generation plus 1 is equal to the current generation 1 minus 1 over 2n, where n is the number of individuals in the population. So Bury in 1956 did a very elegant experiment where he started a bunch of fruit fly populations that had a particular trait. For each of these populations, they had intermediate frequencies of these alleles. And he actually was working with about 100 populations. And over time, he kept track of these populations for this one allele. And he found the allele to be at intermediate frequencies, but then each population became fixed for one allele or the other. And this is a histogram of the number of populations that have a certain allele frequency. And after 19 generations, most of the populations were fixed for either one allele or the other allele. Classic example of genetic drift. There was no type of selection going on. It was all just based on random sampling of each population. And this is what happens in small populations that are not connected by gene flow. You can start off with a bunch of well-mixed heterozygous populations, and as time goes by, they will tend to get fixed for one allele or for the other allele. And so these two populations are completely genetically different from each other, not because of natural selection, but simply due to genetic drift. So genetic drift is most obvious when you have large-scale random effects, such as bottlenecks and founder effects. It's especially noticeable during recolonization events. So a bottleneck is classically illustrated by an actual bottle where you have an equal frequency of blues and yellows in this bottle, and they go through a bottleneck so that only a few individuals from the population survive. Just simply due to randomness, you are going to bias that sample for either yellow or for blue, and when the next population reproduces and grows again, you're going to end up with a population that is much more dominated by one or the other allele. So considering our classroom model, you can have a lot of diversity in the population, and after a bottleneck, the bottleneck will remove all but a few alleles from the population. And we can see this in founder effects. When you have a very diverse population, and when new habitat becomes available, only a few of the individuals will successfully colonize it. And those few individuals will be represented by just a few genotypes, so the new population will be fairly low in genetic diversity and low in heterozygotes. An example of this is seen in tree frogs. Tree frogs have good habitat in desert and grassland. During the last glacial maximum, all of Arizona and New Mexico completely changed their habitats such that they were conifer forests in areas that were completely inhospitable to these tree frogs. When the glaciers receded and the tree frogs recolonized this habitat, the recolonized habitat had very, very low haplotype diversity, very low genetic diversity, indicative that they had recently colonized these areas. So that leads us to this concept of effective population size. We know that populations aren't ideal. Not everyone mates, and mating isn't random. And increasing sampling error, and this increases the sampling error of the available genetic diversity. Effective population size is the size of an ideal population that would exhibit the same degree of genetic drift as is observed in the real one. So this is the population size that evolutionary biologists think about as opposed to ecologists. Ecologists typically think more about the census population size. Evolutionary biologists are concerned about how the population is evolving, and so therefore they are typically concerned with the effective population size. So if you remember the experiment by Bury, those fruit flies were changing in their allele frequencies and becoming fixed for one allele or the other. The rate at which genetic drift occurred in those populations was similar to what you would expect for a population of 9 individuals. If we follow the empirical data and we take that equation that I showed you earlier and outline our expectations for how quickly heterozygosity should be lost, they were losing their genetic diversity at the rate of a population of 9, when in fact the true population sizes were 16. And effective population size is impacted by a lot of different things, including how many males and females are mating in a population. If there's an uneven number of males and females in a population, then effective population size will be reduced. So if we have an even number of males and females mating, it will be close to the actual size of the population. But if there's a smaller number of males, for example, then the population size will be much smaller. Try doing this calculation for an effective population size of 1,000 where there are 400 breeding males and 600 breeding females. Then try doing the effective population size calculation for a population where there are only 10 breeding males and 990 breeding females. You'll find that for the more even number of males and females, the effective population size is 960, which is only a little bit smaller than 1,000. For the very uneven population sizes of males and females, the effective population size is 39, which is a lot smaller than 1,000. So you'll have a much higher rate of genetic drift in this population of 1,000 than the rate of population of genetic drift in this population of 1,000. Bottlenecks and population size fluctuations also affect effective population size. So over multiple generations, the effective population size is the harmonic mean of the census population, of the effective population size over time. So effective population size is the number of generations over 1 over the effective population size at time 1, 1 over the effective population size at time 2, 1 over the effective population size at time 3, etc. So we can calculate effective population size given that harmonic mean formula for census counts of frogs, where the size is 1,000, 1,000, 20, 20, and then 1,000. Try doing this calculation before moving on. And so the number of generations is 5, and we put each value, we take the reciprocal of each value, add those all together, and we get 48.54 individuals as the effective population size. So even though this population has, for the most of the time that we counted it, had an effective population size of 1,000, the bottleneck had a huge effect on the real effective population size and reduced the genetic diversity quite a bit. So bottlenecks will cause allele frequencies to greatly diverge from one another and genetic diversity to be lost. And a real example of a bottleneck can be seen in elephant seals, which had many different genetic types that we know of by measuring bones from middens, from Native American middens between 1000 and 1800. During the peak of hunting, they shrank to a very small population size, and ever since then there's been much less genetic diversity in the population. We can also use ancient DNA to allow us to look backwards in time using coalescent methodology. And by using this ancient DNA, we're actually able to see that the bison effective population size over the last 50,000 years declined right around 20,000 years ago, 15 to 20,000 years ago, which is right when humans arrived in North America. Very interesting, suggesting that humans were involved in population decline of this species. So the biggest takeaway from this lecture is to remember that genetic drift tends to reduce genetic variation within a population because it tends to cause alleles to either go to fixation or to extinction. However, among populations, it tends to increase the amount of genetic variation because it will cause some populations to be fixed for one allele and other populations to become fixed for a different allele. So prior to the 1960s, it was believed that selection was the driving force in the evolutionary process. So if organisms have been under selection for a long time, then there should be very little genetic variation in the natural populations because they should all be fixed for the most advantageous alleles. And remember that selection is another force that removes genetic variation from populations by causing alleles to become either fixed or to go extinct. So the classical school of population genetics believed that polymorphism, genetic polymorphism, is rare in populations, and that mutations are either deleterious or advantageous. And advantageous alleles would be taken to fixation by selection, and deleterious alleles would be immediately lost from the population due to selection. So the overall idea was that mutations are either very good or very bad. There was another school that was involved more in actually measuring genetic variation. They believed that polymorphism in populations is common. But there was no really good idea of how polymorphism in populations would be maintained if selection was the main force at play. The overall idea was that if there is polymorphism in populations, it's maintained by heterozygote advantage or overdominance. So remember that heterozygote advantage selects for two alleles in a population because it selects for the heterozygote that contains both of those alleles. This was an unsatisfactory answer, though, because remember that it's impossible to have more than half of the population as heterozygotes. And if heterozygotes are consistently the most fit individuals, it means that you can only have half of your population under Hardy-Weinberg with a heterozygous genotype and therefore the most fit. And this is about the time that people were starting to actually be able to look directly at genetic variation by taking protein products called allozymes and running them on gels and measuring the actual changes in proteins that occurred in populations. And so this is an example of an allozyme gel where we can see clear differences between these individuals based on what alleles they have at different loci, changes how far these allozymes run on this gel. So then, in 1968, Motu Kimura came up with this idea called the neutral theory. And as often happens in science, it was almost simultaneously proposed by King and Jukes in 1969. Now, the neutral theory is kind of a subtle but very powerful idea, and I'll try to explain it in the next few slides. So recall that a mutation is simply a change in the DNA of a single individual. You don't have a substitution until that mutation becomes fixed in the population. So this is a mutation, a change in one individual at one of their chromosomes. Often that mutation gets lost due to extinction. Occasionally, due to genetic drift or due to selection, that mutation goes to fixation. And in between, we have genetic polymorphism, where we have the allele at some intermediate frequency in the population. Now we can calculate the probability of fixation as based on the initial frequency of the allele, so one individual has it at one chromosome, and the effective population size. And we can also add in the selective advantage or disadvantage if selection is at play. So for a new neutral allele arising in a diploid population of size n, the initial frequency, as we stated at the beginning of the lecture, is 1 over 2n, because there are 2n gene copies in the population. But remember that one allele is always going to go to fixation in a population. So any allele that's in the population also has a 1 out of 2n chance of going to fixation. So when it's brand new, there's only one copy of it in the population, and its probability of going to fixation is same as all the others, assuming neutrality. And it has, therefore, its probability of going to fixation is 1 out of 2n, just like its initial frequency. So if we have a very large population, it will take a long time for any individual allele to go to fixation, and you'll therefore have lots of alleles segregating in the population, kind of waiting for, just kind of bouncing around due to genetic drift, and not going to fixation or extinction in a very large population. So if you have 1 million individuals in your population, and they have a generation time of 2 years, and we're talking about selective loci, the time by which a new allele could go to fixation would be about 8 million years on average. As an aside, if there is a selective advantage, then alleles go to fixation very, very quickly, as we saw in the selection lecture. An allele can go to fixation in the rate of fixation is 4n times s times u. So you add in that selective coefficient, and it causes the alleles to go to fixation very, very quickly. Now, the fixation rate under neutrality, if we're interested in what the substitution rate is, how often we're changing a letter of the DNA from an A to a G, for example, recall that the probability of fixation is 1 over 2n, but the number of new mutations in a population is also related to the effective size. The number of new mutations in a population is 2 times the effective population size times the mutation rate. So if we set these two quantities, if we multiply these two quantities together, the mutation rate is equal to 2ne mu times the probability of fixation gives us the substitution rate. Effective population size drops out of the equation, as does the 2, and we end up learning that the substitution rate is equal to the mutation rate under neutrality. This is quite an amazing finding and very elegant. So, the neutral theory states that the great majority of mutations that are fixed are effectively neutral with respect to fitness and are fixed by genetic drift. And that polymorphism within populations is transient and just due to the presence of neutral alleles that are just drifting around on their way to fixation or loss. So the reason that we find genetic polymorphism in populations is simply due to all these different alleles that are floating around and are either going to become fixed or lost from the population. So, the neutral theory does not de-emphasize the importance of selection. It still says that all adaptive evolution is due to natural selection. But it says that advantageous mutations are rare, and most genetic variation at the molecular level is not selected within a population. So most genetic variation is neutral and therefore only subject to genetic drift. Most substitutions are also not due to selection. So the predictions of neutral theory are that the rate at which mutations are fixed should be higher when there is no selection involved. So we would expect that there would be a higher synonymous mutation rate than non-synonymous mutation rate, because remember that neutral theory predicts that most non-synonymous changes will be deleterious, not advantageous. So we'd expect to see a higher substitution rate in synonymous changes and in areas that don't code for any genetic product, such as in introns and flanking regions. So remember that most mutations in a genome are going to be neutral and have no effect, and the next largest category of mutations are going to be very, very deleterious or lethal. So therefore, we would expect to see a very high rate of synonymous substitutions in a population and a much lower rate of non-synonymous substitutions in a population, since only a very few of those, only a very few of the non-synonymous substitutions are actually beneficial and would become fixed if they're exposed to selection. And this is a figure from one of Kimura's papers that shows that the synonymous mutation rate is much higher on an exponential scale than the non-synonymous substitution rate for genes that actually code for something. The second prediction of neutral theory is that the substitution rate is equal to the mutation rate and that this is independent of population size. And this is a truly amazing result. This means that larger populations will have a higher population mutation rate, but they'll also have a lower probability of fixation. Smaller populations will have few mutations, so they'll have a small effective population size, which will mean fewer mutations, but they will also have a higher probability of fixation. So the neutral substitution rate, just like the mutation rate, is basically constant over time. And this is what leads us to be able to predict a molecular clock. So the rate of substitutions that we see between species is actually related to time. And we can build a phylogeny and count the number of mutations along the phylogeny and add those mutations up and relate those to time. If we know the mutation rate, then we know the substitution rate as well, and we can actually calculate the number of years or number of generations that two species have been separated from each other. And we'll learn more about this overall idea when we talk about phylogenetics. So they were able to calibrate the first genetic clock using the alpha-globin gene and found that the number of years that two lineages have been separated from each other is highly correlated to the number of genetic differences that are seen at this alpha-globin gene. So for example, humans and dogs, the two lineages have been separate for about 80 million years, and they have a relatively small number of genetic changes at this gene. The genetic distance is relatively small compared to the other genetic distances. And we can plot that right here and see that the number of differences between each of these species is highly correlated to the amount of time that they've been separated from each other, thereby showing a clear molecular clock. And we can do this just within mammals and see again a very clear molecular clock where the number of substitutions is highly correlated to the divergence time in years. So with that, the concepts that I'd like you to understand are listed here.